In this lecture, we'll be discussing the dark reactions in plants, also known as the Calvin cycle, and how this leads to carbohydrate synthesis. And the title slide, I think, is a pretty good overview of what you should probably take away from this. I'll be going kind of quickly through this lecture. In this chapter 20 in the book, I don't really expect you to know many of these details. We're just kind of going surface level information on this. But one of the things that's important is that you understand how carbon dioxide is incorporated into sugars and how plants use that to this system to take in carbon dioxide and make carbohydrates, and the general ATP usage of this. Okay, and it happens in 3 phases as shown here, and we'll go through those phases. So this is section chapter 20, I believe that should be section 1. So this is chapter 20 of the book. And the first thing we'll talk about is the carbon dioxide assimilation reactions. Okay, so the products of photosynthesis, ATP, NADPH, these are used by plants to reduce atmospheric carbon dioxide into trioses, or 3-carbon sugars. Right, so as sunlight comes in, the plant gets the energy it needs, and then it can use carbon dioxide plus that energy it's made from photosynthesis to create these 3-carbon sugars. Those can then be used to produce larger sugars like glucose, and that would lead to energy production for the plant, for the cell. You can also Plants could use these to build up larger storage polysaccharides, things like starch or even sucrose, that plants use for storing them or transport. Okay? And then you can also use these 3-carbon sugars for biosynthesis, things like DNA and RNA. Okay? What we'll really focus on here is the assimilation of carbon dioxide into these 3-carbon sugars, not so much of the details on the other pathways, but just know that that's what those are going to be used for. Okay. So there's a couple terms, CO2 assimilation and CO2 fixation. Honestly, I mean, I'm sure there is a difference, but you can kind of think of them as interchangeably. Right. CO2 assimilation is really, I think the book uses this as the process of taking CO2 and putting it into a sugar. And carbon dioxide fixation is really what we're talking about here, is the first step of this process where the enzyme Rubisco takes CO2 and it makes the 3-phosphoglycerate out of it. So technically that's what they mean, but honestly, I think you could use them almost interchangeably. Okay, carbohydrate metabolism in plants is a bit more complex than in our cells, and that's because they have the same machinery that we do in our cells, things like the glycolysis pathway, gluconeogenesis, and the pentose phosphate pathway. But then they also have photosynthesis and the ability to take CO2 from the atmosphere and assimilate it into sugars as well, which we don't have the ability to do. So this assimilation occurs in 3 stages. Okay, so stage 1 is fixation, where you have ribulose 1,5-bisphosphate, right? You have, and the stoichiometry on here is a little bit complex. to make the numbers work to show a net gain here. So it might be a little bit confusing, but we'll try to go through this. Okay, there are 3 molecules of that, and this is a 5-carbon sugar, so that's 15 carbons there. You add 3 molecules of CO2, so that's another 3 carbons. Okay, so in that first step, you get a total of 15 plus 3, or 18 carbons. Okay, and that's reflected in the product here of 3-phosphoglycerate. You have 6 of them, right? It's a 3-carbon sugar, Triose. You have 6 of them, so that's a total of 18. Okay, stage 2 is reduction. This is where you use the energy that was produced in photosynthesis. So 6 ATPs and 6 NADPHs are used, 1 for each of those 3-phosphoglycerates. That's used to reduce the 3-phosphoglycerate into glyceraldehyde 3-phosphate. And if you remember, that is something we talked about in glycolysis, that molecule. Okay. You have 6 of them, okay, 18 carbons. One whole one of those you can kind of think of as being taken out of that cycle, and it can go on to do other things such as glycolysis or creating starch or whatever, okay? And that leaves 5 of those 3-carbon sugars. So 5 times 3 carbons would be 15. That's then converted back into ribulose 1,5-bisphosphate. You're using 3 ATPs to do so, and you get 3 molecules of that 5-carbon sugar. Okay, so you're generating each round a net of 1 3-carbon sugar of this, but the numbers here are a little bit, different, or maybe complicated, I guess is a good word. Okay, so if we look at this in a little bit more detail, right, stage 1, again, this is the fixation stage. This is where we take the carbon dioxide, ribulose 1,5-bisphosphate, and create 3-phosphoglycerate. Right, stage 2, again, this is reduction. We have 3-phosphoglycerate, 6 of them. Use 6 ATPs, 6 NADPHs, and we get 6 glyceraldehyde 3-phosphates. Again, this is shown as like a couple arrows here, as a couple steps. I'm not expecting you to know the exact steps, right? Just know that that is considered the second stage. You reduce, you use that energy you've built up with photosynthesis, and you get out glyceraldehyde 3-phosphate. Okay, and finally, the stage 3, this is what they consider carbon dioxide assimilation, and that's because the 3 carbon dioxides reflected in one of these glyceraldehyde 3-phosphates can then be kind of go out of that cycle and be used for energy production or energy storage. Okay, so it's just a little bit of a kind of a bookkeeping thing. Depending on what the need of the plant is at the time, where that goes, but that's what's considered assimilation. And then the conversion back into your starting ribulose 1,5-bisphosphate with the usage of, again, more ATP. Right, so I included this clicker question. The Calvin cycle incorporates a complex series of reactions that regenerate ribulose 1,5-bisphosphate. What pathway operating in reverse is most similar to this third stage of the Calvin cycle? Right. So, maybe didn't really talk a whole lot about this pathway, and it might be hard to see right now, but it's most closely related to the pentose phosphate pathway, but just operating in a reductive manner instead of an oxidative manner. So if we talk a little bit more about stage 1, the fixation of CO2 into 3-phosphoglycerate, right, there's different types of plants, and so the ones we're going to focus primarily on are what are known as C3 plants. Okay, and C3 just stands for 3 carbons. And those are plants that have the 3 carbon compound 3-phosphoglycerate is the first intermediate in this photosynthesis carbon assimilation. And if you're interested, the types of plants are listed here. Most trees are like this, so I think, typically this is, when you think about photosynthesis, a lot of times you think about trees, and this is the method of carbohydrate production in most of them. Okay. And the enzyme that's responsible for this, I've mentioned it before, its full name is ribulose 1,5-bisphosphate. Okay, remember that's kind of the starting compound we use. And the enzyme, what it does is carboxylase and oxygenase. So that's a very long name, right? But luckily there's this abbreviation for it, and it's called Rubisco. So you can kind of see that, it's an abbreviation. Ribulose is the rub part, bis is bisphosphate, and CO is carboxylase oxygenase. This is really one of the biggest enzymes on the planet. If you just added up, all the mass of the enzyme, there's so many plants that use it that the amount of this on the planet is just enormous. Okay. And it has a very good job, right? It takes CO2 out of the atmosphere and it converts it into sugars. So you can imagine that this would be a very powerful enzyme if you wanted to decrease the amount of CO2 in the atmosphere, how we've just kind of increased over the last couple hundred years the amount of man-made CO2 in the atmosphere. Researchers and engineers are trying to work with this enzyme to create some way of taking that CO2 out. And we'll talk a little bit more later in the lecture of why that's kind of tricky, or one of the reasons why that's tricky, but it's a very intriguing enzyme for biotech. There's 2 forms of it. One is form 1, enzyme from vascular plants, also algae or cyanobacteria. Form 2 is the enzyme of photosynthetic bacteria. Okay. Don't really need to know these structures, but they're just understand. They're, in case you're interested, there's 2 forms of these enzymes. Form 1, you can kind of think of as the predominant form throughout the biosphere. Okay. So a question about Rubisco. Okay. Does Rubisco stand for ribulose 1,5-bisphosphate carboxylase oxygenase? Yes. Is it the most common enzyme in the biosphere? Yes, that as far as we know. Does it catalyze the covalent attachment of CO2 to ribulose 1,5-bisphosphate and cleavage to form 2 molecules of 3-phosphoglycerate? Yes. And is it activated by Rubisco Activase, which overcomes product feedback inhibition? So that one we haven't talked about, but that one is also true. So all of these are true. Okay. Rubisco has a magnesium in the active site. Okay, and the only reason I kind of bring this up is because that's sort of similar to chlorophyll, right? Chlorophyll had magnesium in it, in that porphyrin ring, sort of like a heme group. Rubisco also uses magnesium. So magnesium's a pretty important metal in plant biology. Okay. Active site, what residues coordinate this magnesium, not super important. We'll talk about a couple here in a second, especially this lysine. Right, so this is the mechanism, at least the first stage, this carbon CO2 assimilation or fixation, as the book terms. The things I want to point out, I'm not expecting you to know this mechanism in any detail. What I wanted to point out is this lysine actually has something attached to it, right? A CO2 group on it. And so this is referred to as carbamylated. Okay, so that'll come up here in a second. So it's a modified lysine. Right? The other important residue is a histidine. It's not coordinating the magnesium, but it's going to act in the catalysis of this reaction. Okay? So CO2 comes in, it's coordinated by magnesium. Then you can get via help from this histidine, removing this proton and this electron moving, you can get attack on this carbon and CO2 from this double bond, and that produces an ene diolate intermediate. Okay, kind of similar to some of the enzymes we've seen before. But again, you don't need to know any details on this. Okay. Well, Rubisco is actually inactivated or inactive until it's activated by the enzyme Rubisco activase. And that's the enzyme that puts that CO2 and carbamylates the lysine at the active site. So this enzyme makes it active. Okay. All right. So looking at stage 2 a little bit more detail. This is the conversion of 3-phosphoglycerate into glyceraldehyde 3-phosphate, right? Very similar, but you're reducing this molecule. Okay. And this requires ATP. Some of the enzymes that are used Triose phosphate isomerase and aldolase, enzymes that we have seen before. Okay. The final stage, that's when we regenerate ribulose 1,5-bisphosphate from the Triose phosphates that we've generated. Okay, and this one is actually pretty complicated. Okay, and don't expect you to know any of these details, but it looks a lot like the pentose phosphate pathway. Okay, and so that's where we get the idea of this pentose phosphate pathway in reverse. It's because you're combining glyceraldehyde 3-phosphate and dihydroxyacetone phosphate and creating fructose 1,6-bisphosphate. So it's a 6-carbon, right? You take one of those 6-carbon molecules and combine it with another glyceraldehyde 3-phosphate. In what you get is a 4-carbon and a 5-carbon, right? And so you get this, and that's kind of all that I really want to mention there, but you get this sort of complex way of creating back to a 5-carbon sugar. Okay, don't need to know these details, but just understand that it is complex and it's similar to the pentose phosphate pathway. Okay. So let's talk about the overall bookkeeping. If we go around a circle here, and the circle is what you'd have to do to generate 1 3-carbon sugar, and that's indicated here coming out as glyceraldehyde 3-phosphate. Okay. We start with 3 CO2s. Those are 3 carbons that we're going to gain a 3-carbon sugar from. Okay. What do we need to do that? We need 6 ATPs in that step, 6 NADHs in this step, so that is both stage 2. Okay. And then in that reproduction of the ribulose 1,5-bisphosphate in stage 3, we also need 3 ATPs. Okay, so that's a total of 9 ATPs and 6 NADPHs. Okay, so that's a pretty large energy expenditure. Okay. Not going to ask you, well, I guess a test question could be, when this 3-carbon sugar goes back into glycolysis, how many ATPs can be generated. Okay, so I suppose that, you know, assuming everything's the same as in a mammalian cell glycolysis that we've talked about, you should be able to kind of come to an answer there and find the total number of ATPs generated from that sort of a process. So unlike starch, sucrose is made in the cytosol. Okay, starch is made in the chloroplast, but sucrose is made in the cytosol. So to get these, to do that, you're going to need to move those 3 carbon sugars that you generate in the chloroplast into the cytosol. And how you do that is by a particular antiporter. Okay, one that will you move a 3 carbon sugar out and you move an inorganic phosphate in. Okay, why is that important? Well, it's important because you need those inorganic phosphates to phosphorylate the sugars that you're making, right? And to generate ATP. Okay, and so it's moving the sugar out where it needs to be, moving those phosphates back in so you can keep generating ATP. Okay, and in particular, don't need to know this really, but it's being converted into the dihydroxyacetone phosphate to be moved out. Okay? And there in the cytosol, it can make sucrose. Right? They can't, these phosphorylated carbon molecules can't just go through the membrane because the phosphate group's very highly charged, doesn't go through the membrane. Right? This is coupled, so that antiporter is coupled with one that moves 3-phosphoglycerate back into the stroma and moves out inorganic phosphate. So you have this coupling. So the enzymes in the Calvin cycle are indirectly activated by light. And that probably makes sense, right? When the light is strong, there's lots of light, lots of photosynthesis is going on, you're generating lots of ATP and lots of NADPH. That's when the conditions are great to actually start making sugars, making carbohydrates for energy, primarily for energy storage. So all of these enzymes in the Calvin cycle, the dark reactions, are catalyzed and activated, I should say, by light. Okay. The last thing we'll talk about is photorespiration, and we'll talk a little bit about C4 and CAM plants. Again, I don't want to get too hung up on the details here. Just wanted to present them as they are. There are different types of plants, and they have slightly different methods for assimilating CO2 that are interesting, so I wanted to point them out. Okay, so there are processes that produce CO2. One, mitochondrial respiration, okay, the oxidation of substrates to carbon dioxide, and you convert O2 to H2O. Okay, and that happens, we talked about that in chapter 19. And in plants, so that's what's going on in our cells. But in plants, that also happens. It just happens more so in the dark when photosynthesis is not an option. Okay. Another way that plant cells can produce CO2 is what's known as photorespiration. Okay, so think of it as mitochondrial respiration. You're creating CO2, and you're using O2 to do that. Photorespiration, same thing. You're producing CO2, you're using O2 to do that, but it's being catalyzed, if you will, by light. Okay, and so this is a side reaction of photosynthesis, and it's a pretty costly side reaction. Okay, And why it occurs? Well, it's from the lack of specificity of that enzyme, Rubisco. So that enzyme that in stage 1 that fixes CO2, it's not very specific for CO2 as a substrate. And because of that, about once every 3 or 4 turns of that cycle, an oxygen binds, O2 binds, and it incorporates the oxygen instead of the CO2. And when it does that, it produces 1 molecule of 3-phosphoglycerate, which is good, but you don't have that extra carbon from the CO2, and the other molecule that's produced is this 2-phosphoglycolate. Right? So that glycolate is actually kind of like a byproduct in sort of poisonous to the plant. If that's in too high of a concentration, it's not good. So the plant has to get rid of that. Okay, and so this is, constantly being produced. If you think of that, it's like 25 to 33% of the time you're producing one of these. So that if you didn't have a way to take care of it, it would build up to a pretty high concentration. And so this is really one of the main drawbacks or main factors that inhibits the use of Rubisco to actually use it like in a biotech way to remove CO2 from the atmosphere, is that you have this non-specificity, right? And you can think of it, there's so much more oxygen in the air than there is CO2, that if you tried to just use Rubisco, sort of an engineered Rubisco in this way, you're really going to make a lot more of this phosphoglycolate and not pull out the CO2. So that's what people are working on, trying to how do we engineer Rubisco to be more specific for CO2. Okay. How the plants get rid of this glycolate that's produced is the glycolate pathway. Okay, don't need to know really any specifics of this, but just know that there is a glycolate pathway. It removes this because Rubisco is not specific. It makes glycolate when it uses oxygen instead of CO2, and this is a way the plant can get rid of that. The problem here is if you follow this through, right? First you take that phospho group, the phosphate off of that, and get glycolate. Then you this happens in the peroxisome, but you convert glycolate into glycine. Then in the mitochondria, the glycine is converted to serine, and then you kind of go back through this to get glycerate and back to 3-phosphoglycerate. And what you're doing here is you're using NADH and an ATP here to do that. So one thing I wanted to point out here with glycine, this glycine conversion into serine by glycine decarboxylase and then serine methyltransferase enzymes. I actually work with the glycine decarboxylase enzyme system. And so I just thought that was neat that, plants use this too. And this was kind of in the book. And in fact, plants were kind of the first place this enzyme was studied in great detail because you could get lots more of it than trying to isolate it from human samples. Just a little bit about it as an aside. If you have mutations in any of the proteins in the glycine decarboxylase enzyme system, it inhibits your ability to break down glycine, and glycine's a neurotransmitter, so it builds up in your brain, and you get kind of overstimulation of some of the neurons So you get overstimulation of some of the neurons in the brain, and it can lead to some very bad things. And if you have these mutations, it kind of shows up in the first weeks of life, and it leads to a disease called non-ketotic hyperglycemia, which can, there's varying degrees of it, but usually it's very bad. In the severe cases, these patients don't make any developmental progress their whole life. So, they really can't do anything. So it's a very devastating disease. And my lab at Anschutz is trying to develop therapies that can help this, the metabolic consequences of having this enzyme disabled. So photorespiration, getting back to on topic here, the overall process consumes oxygen, right? That Rubisco is using oxygen, and it's producing CO2 with it. It's not conserving energy, right? Because you have to use ATP to get it back to convert the glycolate back into the 3-glyceraldehyde. Well, there are different types of plants. So we've talked about C3 plants. C4 plants are a little bit different. Okay, the carbon dioxide fixation and Rubisco are separated spatially. So they're in different compartments. And that's to try to get, alleviate the photorespiration problem. Okay. Right, so in C4 plants, they utilize a carbon dioxide assimilation process to minimize that photorespiration. And C4 plants tend to be more tropical plants and crops that are grown in higher light intensity and at higher temperatures than, the average sort of temperate plants. Yeah. Right. So if we look, and again, you're not going to be responsible for these details. I should have probably put an FYI up here on this slide, but don't stress about these details. Just understand that in C4 plants, CO2 assimilation is separated from Rubisco. So how this happens is carbon dioxide is converted into bicarbonate. Okay, so that's carbon dioxide in gas form converted into bicarbonate, which is soluble, right? Then PEP carboxylase converts it into oxaloacetate. So we've seen that enzyme before, right? We've seen oxaloacetate, malate before. Okay, right? So that happens in a special type of cell. Okay, the malate kind of gets shuttled back into another area in a connected chamber, and that's where you can get from malate you get carbon dioxide back from that, and then that's where Rubisco is, and then you get the Calvin cycle, the normal sort of Calvin cycle that we've talked about in C3 plants. Okay, all this does is it kind of keeps this in an environment where there's little oxygen around to give photorespiration. Yeah, I've included these steps just in sort of detail, but again, not going to really need to know them for this class. Okay. Well, as you can imagine, the process of doing this, we've seen that there was extra steps involved there, right? And it was kind of a cycle. So you can imagine that you're probably not going to, you get a benefit from doing this, right? You're keeping oxygen away from Rubisco, but you have all these extra steps, and you can, and it's a cycle, right? You're not just using up a bunch of something and not regenerating it. It is a cycle. So you can imagine that there's probably going to be a cost to doing this, and there is. C4 plants that have CO2 assimilation in this fashion need 5 ATP molecules to assimilate 1 molecule of CO2, right? Compare that in C3 plants, they only need 3 molecules of ATP, right? So that's 2 extra ATP molecules, right? You're almost doubling the amount of ATP needed to do this. So there is a pretty high cost of doing that, but this is, you can think of the benefit. One, you don't have to spend the ATP to get rid of glycolate, so that's not really factored into this. And then the other factor is that these are warmer climate plants, maybe more tropical. They get more light, they get more heat, right? They can produce more ATP via photosynthesis, so they can use more ATP to fix CO2. Right, the last type of plant that we will talk about are CAM plants. Okay, and CAM plants are the C4 plants we talked about are spatially separated, the CO2 capture and Rubisco. In CAM plants, they are temporally separated, so timing-wise, they're different. So how that works is they fix CO2 into malate in the dark, so at night, okay? And they store it until daylight, and then in daylight, the malate serves as the source of CO2, just like in C4 plants, right, for Rubisco. But these are, instead of spatially being separated out, they're temporally. Okay? And the types of plants that do this are really, you can think of them as, more like desert plants, things like succulents and cactus, and also, interestingly enough, pineapple. Okay? What this does is it reduces the loss of water vapor through the stomata, and we really don't talk about why that is, but just sort of remember, if that helps you remember CAM plants. Not really going to need to know any of these details, but just kind of interesting. Okay, and the book gives this table of the C3 plants, C4 plants, and CAM plants. Not going to need to know really any of this, but just for if you're interested, some of you might be into botany or whatever, the types of plants, not going to ask that, but it's interesting to see, the environment these tend to grow in, right? So the plants that are sort of temperate or room temperature type plants are C3 plants. A little bit warmer and drier C4, and then the extremely dry and warm are the CAM plants.